So, hi everybody. So, my name is Chengsheng. I am a postdoc at Rutgers University. So, it's very happy for me to be here, although I can't see you guys, but I'm happy to be here to talk to you about the function analysis of the microbes and the microbiomes and their relationship to the environment. So, I want to start from this quote of Darwin in his most famous book. So he says that I was much struck how entirely vague and arbitrary is the distinction between species and varieties. So basically what he says here is that to give different organisms different names correctly is not an easy task. So indeed, if we look at this example of 4 different birds living on the same island, so you can see that they bear to an extent certain differences in terms of their beaks and their heads according to the environment that they live in. But however, these differences are subtle. Unless you are an expert who are very into this specific kind of bird, by just looking at the picture, it is not very easy to tell like which one is which. And now imagine when we get down to bacteria, those tiny little bugs, we can't even see them. So this issue becomes even more complex. So currently, the gold standard to tell whether 2 bacteria of the same species or not is using this technique, which is called DNA-DNA hybridization. So the idea is that you take the DNA from both bacteria, you denature them into a single strand, and then mix them together and let them reassociate. And then you count the rate of how much of these heterostranded DNA are there. What is the fraction of those? And the current rule of thumb is that if there are more than 70%, then you can safely say that these 2 bacteria are of the same species. But however, although this technique is very accurate, but it's experimentally very demanding. So it is challenging that people start to seek for another marker which is easier to use. And the answer that we have found is 16S RNA. If you have been into this field, you must have heard about this gene. And it's always associated with a magical number, which is 97% sequence identity. So the rule of thumb is that if 2 bacteria share more than 97% sequence identity of their 16S RNA, then they are of the same species. It is quite widely known and practically used in this field. However, it's just not correct. So what do I mean here? So if we look at this picture, the x-axis is this DNA hybridization rate, and the y-axis is a 16S RNA sequence identity. And each dot in this figure is a pair of organisms. So if we look at all the organism pairs have less than 97% 16S RNA sequence identity, we can see that without an exception, all of them belong to different species. But however, if we look at above 97%, we both have the same species organism pairs as well as different species organism pairs. So this rule of thumb is not really accurate in reality. So in order to quantify, how accurate it is really, so here is something that we did. So we collected 1,400 bacterial genomes from NCBI, which covers the main domain of bacteria. And then I compared the 16S sequence identity across every single pair of these organisms and used that to predict whether these 2 organisms are of the same species or not, and measure the performance based on different cutoff I chose. So for example, if we use this magic number of 97%, so for organism pairs that share more than 97% identity, there are more than 55% of organisms actually of different species. And then for those have less than 97% identity, actually more than 15% of them are of the same species. So what this says basically is that if you're using that famous rule of thumb to identify 2 bacteria to belong to the same species using more than 97% of 16S RNA identity, then you are more than half of the time will make a mistake. So people started to know this and then proposed that, okay, 97% is not stringent enough. Let's do 98%, 99%. So, but in reality here, even if we use 100%, just completely identical 16S RNA sequence, it still does not guarantee the two organisms are of the same species. The precision is actually just about 75%, like 3 out of 4 are correct. So by now, I hope that I have convinced you that 16S RNA is not the best tool to use to identify same species. And this is just an example. The 100% I identity doesn't really make BAF a true BITES. So if we cannot use 16S RNA, then what can we use instead? So we could actually measure all of the different features of certain bacteria, for example, like its morphology and physiology through all those biochemical tests, and then just to find out all the details of what a bacterium is like, and then you can comparing that to the other bacterium and see if they are of the same species. However, as just you can tell from my description, it requires a lot of work, and it's almost impossible to measure everything. So is there a shortcut? So the shortcut lies in the molecular function, which is encoded in the organism genomes, which reflects the morphology and the physiology of that bacterium. So if we were to use this function, molecular function as a measure, then there are also some issues with it. The first thing is that a lot of the proteins in bacterial genomes are not yet annotated. So here I collected the same 1,400 bacteria, it's of the same data set, which contains 4.2 million proteins. So we can see that on average, about a third of those proteins are not annotated. We don't know what function they perform. And in some organisms, for example, the one at the very right, it can be as high as more than 80% of the protein in this bacterium have no annotation at all. We have no clue what they are doing. So yes, no annotation is a problem. But then how about those proteins that we do have annotation with? There is also another problem, which is mis-annotation. So in this paper by Alexander Schnome from 2009, so they have shown that in the big basic public databases like NCBI, CAG, or SwissProt, there's from 5% to 63% of mis-annotation in those databases. And this number is really scary. And it's more scary that once the errors get into this database, it stays there. It's very hard to get them out. And this error just propagates to generate more errors. So it just makes the situation worse and worse. But then, why would we have this mis-annotation in the first place? So the culprit lies in the blind faith of people in the homology by numbers. So what do I mean by that? So if you have a protein sequence with an unknown function and you want to figure out what this protein is doing, so the most common practice is that you just take it to NCBI and you blast it against some reference database, and then you look through the hits and find out what this protein is doing. So I give you some example here. So if you focus on the first two lines here, so you can see that the blast E value are very decent, which indicating that these proteins should share the same function, while in reality, they're actually not of the same function. So what I'm saying that this function transfer by homology is not always correct. So is there another way or a more reliable way to measure whether two proteins are of the same function? Yes, there is. So this is developed by the advisor of my advisor, which is who is Dr. Brooker Roast in 2002. He established this so-called HSSP metric. So the idea is that, first of all, you gather a bunch of proteins from which their function annotation are experimentally verified, which means that they are correct and we can believe in them. And then you do pairwise alignment for all of them. So you end up with a pair of protein, like alignment of proteins of the same function, and you also have alignment of proteins with different function. And then you map all of those alignments into this 2D space of alignment length against sequence identity. And then you train a curve to separate those same function alignment from the different function alignment. And then once the curve is trained, then the alignment above the curve will be predicted to have the same function, and the alignment beneath the curve will be predicted to have different function. And the distance from the alignment to the curve indicating the reliability of such prediction. So now with this new tool, and let's revisit the example that I just showed you guys, you can see that now if I calculate the HSSP score of those two pairwise alignment, the HSSP is correctly giving me the information that is a minus value, which indicating that they are of the different function. So the HSSP is a more reliable way to measure whether two proteins of the same function or not than BLAST. So with this tool, I was able to actually calculate the HSSP score for all of the pairs in the 4.2 million proteins. And then I put all those 4.2 million proteins into this big network. So every two protein is connected by their HSSP score or distance. So in this, you can assume that the proteins with the similar function will cluster together, which I can identify later as functional group. A functional group are actually just protein clusters that contain protein members sharing the same function measured by HSSP. So once I have those, so this is a flowchart of the whole procedure. So once I have identified those protein clusters, what I can do is that I can map each bacteria to a list of molecular functions that its genome contains. So for example, both the blue and the red bug has function 1 and the function 2. And the function 4 is unique to the red bug. So with this, I can further calculate the functional similarity between every pair of bacteria by just comparing the similarity between these function vectors. So if I use that similarity as a new metric to identify if two bacteria of the same species or not, here you can see a significant and obvious performance gain comparing to using 16S RNA. But however, it's still not great because you can still only retrieve like around 70% of precision. So then what, right? So we take the idea of like, all right, if the pairwise alignment or the pairwise comparison of bacteria does not really work, then let's try the network. So now we can actually put all of the bacteria into a big network and connect them by the functional similarity. In the same notion, the bacteria that are functionally more similar tend to cluster together as well. So if I do this to all the 1,400 bacteria in my data set, we get a network somewhat like this, like a fish. It's a fish network. So here in this layout of the network, all the organism nodes are colored by the phyla and the major classes. So we can compare this organism clustering to the taxonomy, and we can see that this network, which we call fusion. So this fusion network actually reflects taxonomy. But however, only to an extent, because here, if I were to use computational methods to identify these explicit and strong subnetwork within this fusion network, by saying subnetwork, I mean that the subnetwork have very strong within network connections and very weak across have very strong within subnetwork connections and a very weak across subnetwork connections. So if I were to identify these subnetworks, and we can see that they do not entirely overlap with the current taxonomy. So why is that? The subnetwork is totally driven by the function similarity, but the current taxonomy is not, because it is somewhat arbitrary, like what I mentioned in the beginning by Darwin. So I'll give you an example here. So at the top, these two Borrelia species, they are functionally more than 99% identical. But however, they cause different subtype of diseases, and we just decided to put them into different species. And at the bottom, this Clostridium botulinum strains. So they are functionally very different, less than 40% identical from one another. But however, because they all cause the same type of disease, we put them just in the same bin in the taxonomy. So this is very arbitrary, just like the bird example, unless you're an expert that spend your life studying these specific bacteria, you will not be able to know what is going on. And this is actually also almost impossible, made it almost impossible to process like in this large-scale computational analysis. So I will leave an open question here. So do you think the current taxonomy needs some modification? How would you address this contradiction here? I would be happy to hear your thoughts. So here, I'll give you another example showing all right, so how function is affected by the environment. So these are, this is a network of 40 cyanobacteria. And I want you to focus on the nodes with the cyan ring. Those are the Synechococcus, which is a genus of cyanobacteria. So you can see that those marine Synechococcus are functionally very different from the freshwater Synechococcus. And they are actually functionally very similar to another marine cyanobacteria, which is the Prochlorococcus. Those are the ones, the nodes with the yellow ring. So here we see a clear signal of how microbial function is driven by environment. And the current taxonomy is not seen, cannot see this change or this difference. So in order to make usage of our data, we built FusionDB, which is an online database which stores all of those 1,400 bacteria information that we calculated, the function and the environment information. So in FusionDB, we allow you actually to submit your organism of interest, submit genome to us, and then what we will do is that we're going to map it to the Fusion data. So once the mapping is done, you're going to see a result page like this. So first, your organism will be mapped to all the molecular functions that we defined. So remember those protein clusters in the big network. So we're going to first map your organisms into the list of functions. And then we will calculate for the functional similarity between your query organism against every single reference organism in our database. And also, as you can see here in this picture, each reference organism is associated with environmental metadata, which includes like temperature, oxygen, and habitat. So you can further select the organism of your interest and using those and your query organism together to create these network visualizations on our database. And you can color them by taxonomy, by environment data, to try to find the patterns and why, to find out what is the reason to cause this function similarity between these organisms. So that is so much of the genome functional annotation. And I will be moving on to the microbiome. So basically, microbiome is just a community of bacteria that live in certain environments. So instead of looking at one bug, now we are looking at a group of bugs. So there are multiple ways to study them, but one of the most commonly used one is using metagenome, which is a sequence all of the genetic material from all of the bugs in this environment. And after that, we get these short DNA fragments, which we call reads. So there's one way to assemble the reads into contig and reconstruct the whole genome. So I'm not going to talk about those today. I'll be talking about directly annotate function from the reads. So there are already some resources dedicated to that. For example, mgrest is quite a famous one. So it annotates the function directly from the reads. So how it does is that, so how does it do it? So it has a reference database, contains full-length proteins with function annotation. And then it will just map the reads to these reference proteins. And then if it matches, then the function of the reference protein is transferred to these reads. But the problem with that is because these are super short alignments, therefore you have a lot of unspecific binding, which means that you end up with a lot of false positives. And another problem is, again, as I mentioned before, the annotation quality of the reference database is questionable. So here you can see that the number, so here in this chart, the bar on the very left is the overall number of proteins in SwissProt. So SwissProt contains only manually curated proteins, manually curated with proteins with functions are manually curated. And then among all of those, how many proteins that we actually have generated and see it in lab? There's only less than 6,000 proteins that we have seen it in lab. So that is the existence evidence at the protein level. So out of 500,000, around 500,000 of the manually curated proteins, only 6,000 proteins we have actually seen the protein. The other ones, we only know the sequence. We have never seen the protein itself. And among these 6,000 proteins, there are actually even a smaller fraction, which is more, fewer than 3,000 protein sequences, whose functions are actually experimentally verified. So we are talking about a very tiny core set here that we have a high confidence in their functional annotation. So we use this set, and what we do is that we use the same idea to train a curve to separate this same function alignment from the different function alignment. But however, here, the same function are the however, here, the alignments are not protein-protein alignment anymore, but the short reads align to the full-length proteins. And once we train the curve that separates those same function alignments from the different function alignments, same idea, we can calculate this. Now we call this new metric phasor. We can calculate this a phasor distance, which indicates the reliability of the function predictions as well. So we compared phasor to SciBlast in terms of the performance, like how well can we annotate functions. So we can see that the phasor outperforms SciBlast e-value by a lot. With phasor, you can actually reach around 90% precision, which is amazing in terms of annotating the short reads. And we also created an online service, which is called MyPhasor, which is available at the website here. So in MyPhasor, we accept your submission of your metagenomes, and we will annotate it for you using the phasor algorithm against that gold standard database where every single protein, for every single protein, its functional annotation is experimentally verified. So once your job is processed, the result looks something like this. So on the top page, there are some statistics and descriptions of the job, and the bottom is the read abundance table. So you have, like, each line indicates one function. So here, all the functions are reliable because they are experimentally verified. And then you have the number of reads that mapped to this function. So the idea is, with MyPhasor, we can turn your metagenomes into functional profiles. Looks like this. And from there, we can do actually a lot of different analysis. So I'm going to give you some examples of what we have done on different samples. The first one I want to show you here is actually based on the BP oil spill sand data. So I'm sure you heard about this. It's very nasty. But however, for this group, they actually had the sand data before oil, during the oil phase, and then have the sand sample like after the oil has been degraded, so the post-oil phase. So we analyzed this data set with my phaser and then use NMDS, which is a visualization method, to compare the functional similarity between these metagenomes in the data set. So you can see it's actually pretty well separated between different phases. But what is more interesting here is actually you can see that this from the pre-oil to oil phase and then to post-oil phase, it's kind of showing the sand microbial community is recovering from this contamination. And another example I'm showing you here are human data. So which is, there are 2 patients with Crohn's disease and their healthy relatives. So what you can see here is that for these 2 patients, so we have their gut microbiome samples. So their gut microbiome are actually functionally very different from their healthy relatives. And which is more interesting is that even between these 2 patients, their gut microbiome functions are also very different, which indicate possible different mechanism of the Crohn's disease, pathogenicity, or how that reacts with the gut microbiome, so on. And this is also, So this is also done by NMDS. So from our experience, NMDS, this visualization method is very good for small sample size. But however, for big sample size, like t-SNE embedding is more suitable. So what I'm showing you here, the last example is that these are from the city subways. The microbiomes are sampled from the city subways from various cities from the world. Okay. So for example, the green ones in this chart are sampled from subways in New York City. And then the cyan ones are sampled from Porto. And then the pink ones, the magenta ones, are sampled from Tokyo, right? So what you can see is that there is a high functional dissimilarity for the New York City samples. They actually scatter around and just everywhere. But however, if you look at the Porto sample and Tokyo sample, they are functionally more consistent to each other. So there are a lot of interesting that you can do with this. Because I'm out of time, I will not go into any further. So here I want to give some credits and acknowledgements to people who are also behind this. So Dr. Machlich, Yannick Machlich, which is on the bottom left corner. So he is also a co-author of the FusionDB. And Dr. Max Miller, which is the guy in the middle on the top. So he is a co-author and also for the MyPhaser. So we are now currently updating these 2 tools. So if you have any idea or any suggestions, please feel free to shoot me an email. I'll be very happy to hear them. And on the right is a lab. So on top is the advisor, so Dr. Jana Bromberg, and then we have our lab mates. So thank you very much.